Hi guys, my name is Nathaly Santibanez. I am a senior and biology major at Oglethorpe University. So today I'll be talking about a behavioral assessment of Ts65Dn mice, a putative Down syndrome model. So the reason why I chose this topic was because I don't think that there's much attention or we don't really hear much about research on Down syndrome, other than maybe genetics, but nothing on really treatments or trying to find out more on Down syndrome. Another reason why was because when I was in high school, I worked a lot with students that had Down syndrome, and I would volunteer, and we would always try to get them to feel included with us, to not make them feel like outcast, and show them that they could do the same things that we did, kind of like we did an Olympics event for them to show them that they could still do sports like us and stuff like that. In no way, I don't think that there should be a physical appearance change with patients that have Down syndrome, but I think that research could help with brain regions and defects with learning and memory for kids that have Down syndrome to get a regular education. Um, Overall, Down syndrome doesn't have a cure, but I do believe that early treatment programs can improve skills. And I think this paper does a great job with seeing, trying to find a model to find, to do further study and further research. So the outline today, I'll first be talking about the background and purpose, then the hypothesis, the experimental setup, results, conclusion, take-home message, and then the resources. So what is Down syndrome? Down syndrome is a genetic condition caused by an extra genetic material from the chromosome 21 resulting from improper cell division. So there are developmental and intellectual impairments that are caused by the genetic chromosome 21 abnormality. And we see that Down syndrome is characterized by distinctive facial appearance, intellectual incapacity, and developmental delays. And they also have a possibility with thyroid and heart illness as well. So, for the background and purpose, so the authors wanted to find a mouse model that helped with their study of Down syndrome. So, they wanted to find another model because the authors proposed the TS16 mouse would be a model for Down syndrome. However, they were limited to their results. And they were limited because the TS16 mice did not survive the prenatal period. However, the TS65Dn mice became a next possible model because they lived to adulthood and they revealed a developmental delay early in life. So that would show evidence like with their lower size prior to feeding, hyperactivity, and tumors. So they had several symptoms of the disease Down syndrome, like I said, with developmental delay and cognitive impairment. So we could relate it to Down syndrome. And so they hypothesized that the TS69DN mice are a good model for Down syndrome due to them only having the distal section of mouse chromosome 16, which is equivalent to a major part of the Down syndrome region of human chromosome 21. So they had 5 experiments, which I haven't seen so many experiments in a paper before. It was a lot to take in. But for their experiments, they did a sensory motor task, exploration, locomotor activity, emotional reactivity, and spatial learning navigation. So for their experiments, they wanted to do behavioral tests, and they had 16 TSDN mice and 16 control mice. And so the control mice were also litter mates. So what that means is that they were all born in the same litter. And um, so they did the 5 experiments, but the experiments I'll be focusing on are 3 of them, which are the sensory motor task, the exploration, and spatial learning navigation. I think these 3 showed really good evidence to why the Ts65Dn mice would be a great model. And I think it showed clearly in the results. So to start with the sensory motor task, this is the progress of receiving sensory information and producing a response. So researchers have proposed a range of sensory motor experiments to explore brain functions such as multisensory integration, sensory learning, and motor control. So with the sensory motor task, they did 6 in total, but the 4 that I'll be talking about is the visual placing, the wooden rod, wire rod, and prehensility. So overall, they wanted to see if the sensory motor task, they wanted to see if the mice had reflexes. And so with the visual placing, they measured the response to extend the forepaws when the animal was gently lowered by the tail towards the surface, towards the flat surface. So you can see that in the bottom left picture, you can see that the mice is trying to expand their forearms. And then we'll talk about equilibrium, and that's when the animals were responding, were placed in the middle of a high horizontal rod, and the delay to fall off the rod was measured. And then lastly, we'll talk about the wooden, wire, and prehensility. So in trials 1 and 2, they used the wooden rod, and then in trials 3 to 4, they used the wire rod. And they wanted to see the capacity of the animal to remain suspended by the forearms clasped around the horizontal bar. And they tested this in 5-second trials. They did 3 5-second trials. And so on the left, we can see that the mice is trying to stay suspended as well. So as for the results, it was good results because their sensory motor tasks were not different. So there was no difference in the control mice and the Ts65Dn mice. So for the visual placing, we can see that they scored, the Ts65Dn mice scored a 3, which indicated that the animals extended their forearms in all 3 trials. And the equilibrium represented the time spent on the corresponding rod. And then the prehensility score of 3 indicated that the animals remained suspended by their forearms during the 3 5-second trials. So there wasn't any differences in the visual placing wooden rod, a little bit in the wire rod, but not a huge difference, and the prehensility also did not have a difference at all. So this shows that the mice, the Ts65Dn mice, don't have a difference in sensory motor task in the way that they can still they can still have reflexes, and they don't have a difference in, feeling like us and stuff like that. So next is exploration. And when I think of exploration, I think of curiosity. So they want to, always want to find more. You always want to see what's around, and just trying to be curious about what they have there in the experiment. So for the exploration, during the light phase of the LD, cycle, they had all 32 mice evaluated for exploration in a whole board. So this was pretty interesting. They had a white wooden box and they had 4 holes in the floor and they were all evenly spaced. And for 5 minutes, they tried to see the difference in number of head dips and the time spent head dipping in each hole. So they would see if the Ts65Dn mice would spend more time or they would be more curious in head dips and do more head dips than a control mice. So as for the results, you can see that there is a big difference in exploration. So the Ts65Dn mice had a big difference in head dips from 43.4 to 61.0. And for the time of head dipping, it was a huge difference as well from 66.1 to 96.9. And so that just goes to show that a Ts65Dn mice is more curious. They want to know more and they try to find what to, like, they try to find new things and they spend more time looking at what they see. So that was the result for that. And the control was on the left and the Ts65Dn was on the right. So the last experiment I'll be talking about is spatial learning navigation. So spatial learning navigation is helped with the outside of the medial temporal lobes and structures that provide extra spatial coding to long-term memory, decision-making, and executive function. So for this experiment, they did something really unique, which I've never heard about before. So for over the course of 5 days, they had 9 control mice and 9 Ts65Dn mice, and they did their place learning in a swimming water maze. So for 4 trial sessions separated in days, the mice were placed in each experiment at one of the 4 starting points. So they had a north, south, east, and west. And the swimming pool was filled with milk opaque water. And I'll explain why they did that in a minute. So the animals were free to swim until they found a submerged platform in a set spot. So basically, they had a box and whatever spot they put it in, and the swimming pool was opaque so that the mice couldn't see the platform so easily. They would try to swim and find their way to the platform. And so this has to do with memory. So they would see how much time they spent and the distance they traveled until they got to the platform. And so another thing to add was that if the mice didn't discover the platform within 60 seconds, they were placed on it for 30 seconds to see if within those 30 seconds, they have it in their brain where it's at, they remember, and then come back in a shorter time period. So this was a unique experiment, which I thought was pretty cool to see how the mice would remember or find their way to the platform. And so you can see that in the little picture where the mice is trying to remember where the cheese is and trying to find its way. So yeah. So as for the results, there was a big difference. So the triangles on the graph are the Ts65Dn mice, and the circles are the control mice. And so for the distance, we'll start. You see that the Ts65Dn mice traveled a longer distance. So they would travel around like 500 in the first session, and they would go down to 200. But there's a big difference with the control mice. As you can see in the first session, they started off around the same period, 400 to 500 centimeters, but they remembered in the second session, so they didn't travel as much in the second session to third and fourth. As for the TS65DN mice, they didn't really have a big decrease, and so they didn't really remember that well, and their distance traveled wasn't as low as the control. You can see there's a big difference there. In session 2, there's around a 200 centimeter difference. As for the time, the TS65DN mice also had a longer time than a control mice. So for the first session, you can see it was about 70% time, and the control mice was a 60% time, and the control mice had a big difference in their time. It dropped to around 30%, and the TS65DN mice stayed within like the 60%. So it didn't have a big decrease, and it would still take them long to find the platform. So this shows that the TS65DN mice didn't find the platform in a significant time, and they would have to be taken back to remember where the platform was. Like I said before in their experiment, if they didn't discover the platform within 60 seconds, they would place them on it for 30 seconds and then run it over again. So the TS65DN mice took a longer time, and they traveled a longer distance. So this goes to show that the control mice had a better memory and cognitive learning as average, but the TS65DN mice had a lower than average. So in conclusion, the TS65DN mice supported the author's hypothesis and showed that there are a significant mouse model for Down syndrome. And this is great news because I think we can see in the results that the TS65DN mice had a greater exploration, a mild impairment in the spatial learning activity, and they had no sensory motor abnormalities. So like I said, they're as normal as us with sensory motor reflexes. They can still do the same things as us physically. As for their exploration, they have more curiosity than us. And as for learning, they have a less, their memory is less than average, where they have not so much they have a short memory loss or long memory loss. So the take home message would be that since TS65DN mice are found to be a perfect Down syndrome model, they can use TS65DN mice for preclinical assessments of drugs for cognition and Down syndrome. So like I said before, I think this would be a great way to find treatment programs to improve skills and to find further studies in ways that they can still get an education like us and get a further education like us and still feel the same and get the same opportunities. So thank you guys. These are my resources, and it was really fun to do this research and read this topic. I don't think I've heard much on it. And so it was pretty cool to see all the experiments that I haven't heard of before. And I hope to hear more about it in the future. And thank you guys.